On behalf of everyone at the Center for Brain Health, welcome to Frontiers of Brain Health, where we take a deep dive into the most exciting new developments in brain research. I'm Dr. Jeremy Gray. I'm the Director of Information Technology here at the Center, and I also have a background in cognitive neuroscience. I was a professor for about a dozen years and have publications doing human imaging. I'm really excited today to see a deeper dive, to go down into the real neuroscience level. We were talking earlier, and I was saying we used to talk about having a view from the cheap seats, like a baseball game where you see way up high and kind of tell something interesting is happening down there. But what's really going on, the real nitty-gritty is a whole nother level. I'm just delighted and really interested to hear what you have to say today. We'll have a few minutes at the end to answer questions. So for the virtual audience, please use the Q&A function. And for our in-person audience, please have your brief questions ready to ask. We'll respond to as many as we can at the end. As a reminder, this presentation is recorded. For those of you who don't know about us, the Center for Brain Health is a cognitive neuroscience research center at the University of Texas at Dallas. For the past 3 decades, we've been leading the scientific quest to measure and improve brain health across the lifespan. Because here is the bottom line. Optimal brain performance is the new frontier of human potential. Now I would like to introduce our speaker. Dr. Nan Li received her PhD in biomedical engineering from the Johns Hopkins University School of Medicine. As a postdoctoral associate at MIT, her focus was on brain circuitry using novel molecular fMRI techniques with particular emphasis on the functions of the neuromodulator dopamine, including a first author publication in the journal Nature. Among her many honors, she was a McGovern Rising Star at MIT in 2020. She's the recipient of the Harvard Chinese Life Sciences Distinguished Research Award, the Stanley Fan Research Fellowship, Women in Bio Robbie Melton Scholarship Award, the UT System Rising Stars Award, there's a trend here, and the endowed Thomas O. Hicks Scholar in Medical Research. As an assistant professor in the Advanced Imaging Research Center at the Peter O'Donnell Jr. Brain Institute next door at UT Southwestern Medical Center, she's working to develop novel whole brain MRI imaging methods to integrate molecular and systems neuroscience while solving brain science problems. She hopes her work will broaden our capacity for functional brain imaging to better understand fundamental mechanisms and develop treatments for better brain health. It's now my pleasure to welcome Dr. Nan Li.
Thank you so much for this wonderful introduction. And also thank you for the invitation. It's really my pleasure to give a talk today here. And thanks for everyone to come. Okay, so I'll start. So you can ask questions in the middle of my presentation. So I'm going to talk about something different, a new technology we developed for studying the brain functions using fMRI. Before I start, as Jeremy mentioned, so my lab is just next door. We should talk more often. So next door, there's this cluster of small animal scanners, 3T, 7T, 9.4 Tesla. So my lab is using the 9.4 Tesla and 7 Tesla small animal scanners for functional MRI methods development. And 2 directions. One is we want to build tools that help us to see more clear what exactly happening in the circuits and molecular levels. And we push everything to the awake studies, awake study for the mouse or rat brain. And using multi-scale measurements, not just MRI, but also calcium imaging, dopamine imaging using optical sensors to cortical levels and cellular levels. Cannot see it? Okay, that's fine. So they're supposed to be cells. And we use all those tools to understand the brain functions, especially in the reward learning and also the circadian rhythm interaction with reward system. Currently, our lab has mainly 4 people working with me. Very lucky to have them. Shuapeng Ma's work will be mentioned today, and then Ray Yang is students, grad students, and Minjang Kim and Yang Yang are working on the reward learning part. So hopefully we'll report those research later. Okay. So talk about the brain functions and the tools. Normally, we think about the brain as a computation center, right? So all the AI tools developed and popular these days, and it's all focused on how this machine is computing information. But actually, how our brain works is not just computation, and how we make the computation possible is by, I will call it this color-coded circuits. All the circuits has different colors based on their own properties, the genetic profiles or neurotransmitters, neuropeptides they are releasing, receptors, all the molecular levels of information, and make them behave differently. And if you have disorders in different circuits, the behavior will review differently, and the computation will also alter based on the neurotransmitter dynamics release or information myelin axon transformation. All those synaptic transmission matters. Right, so how do we actually learn brain function? By imaging. All those informations are critical, and we are pushing more high resolution and more specificity. In the optical field, 2 decades are crazily going on is because of the genetically encodable sensors. They are able to do the whole brain calcium imaging in zebrafish. And this is an example. So you can see which brain areas actually has calcium activity fire up, and then which areas is propagating. This is like a dream for us for functional MRI. Functional MRI is wonderful. It's the only tool that we can use for whole brain study in human subject. There's no other tools to do it these days. But what I'm gonna show you more is when we want to see more. Let's say here is calcium signals, and I also mentioned there are neurotransmitters, different type of neurons. When we want to see more, our fMRI methods are limited. We cannot see the details, but using genetically encodable sensors, if there is something similar as optical sensors in MRI, that will be helpful to review this multidimensional information. So as I mentioned, right, so both fMRI is limited, but still powerful. And there's a bunch of people in MRI field trying this molecular fMRI strategies. How can we review molecular information? But it's really challenging is we want to make some sensor that's sensitive to certain type of targets, right? If I want to make a calcium sensor, it has to be calcium sensitive, and then has to be specific. It has to be reversible. If calcium is in flux here, when calcium is gone, I should be reset to the baseline. And it need to generate enough MRI signals. If there's not enough signals, this level of signal change is not detectable, it's impossible to do the imaging. All right, so it's very challenging. And then our lab proposed and then now is continue this trajectory is doing hemogenetic fMRI. We used both fMRI's fundamental mechanisms, and then we link it to molecular events. And why do we do this? Why do we use the hemodynamic fMRI as a reporting system? Let's dive into why both fMRI works. Back to 1990s, people do. I want to use this slides also emphasize the animal research and then human research are tightly linked, and we can promote the development in this two field. When in 1990s, Ogawa in the fMRI in Minnesota, when he was doing rat study, and he supplied different type of oxygen to the rat. When he supplied a mixture of oxygen and carbon dioxide, his rat MRI image is much brighter than the 100% oxygen ones. And the reason is this carbon dioxide, 10% of carbon dioxide actually caused visual dilation, the whole brain visual dilation, increased the whole T2 star MRI signals. And then in human study, the 2 years later, they reported when a human subject has this visual stimulation in the visual cortex, they observed the same sequence, same contrast, and synchronized signal increase when the light is presented. So visual dilation is observed hemodynamic signals, neuronal correlated based on the human studies. So these two signals are actually linked and they're very potent. This level of a signal, if you do molecular fMRI, you got Nobel Prize immediately. So this is repeatable and even use 1.5 Tesla. Right, these days we have much better scanners and higher resolutions, but this sensitivity and the level of signal change is crazy impressive. The reason is we have blood vessels everywhere in our brain. This is like wonderful report system for us to develop imaging methods in the brain. In human brain, this was collected by Nan Li, another PI in the Advanced Imaging Center across the street. And human subject, animal subject, they even did immunostaining to stain the endothelial cells in mouse brain. You can see how dense those blood vessels are. It's those blood vessels that report the bold fMRI signal we are recording in all those human fMRI studies. So, and then they are also linked to neuroactivities. I'm not going through very details, but the key message is we have different type of cells in this very dense blood vessel structure across our brains. And those blood vessel cells are actually responding to multi-type of cells, neurons directly, and the astrocytes also. There are so serotonin, acetylcholine, those are neuron peptide, neurotransmitters, neuromodulators. They also function directly to the blood vessels. Right. So our both signals are reflecting all those mixtures of information. And once those things happen, our blood vessel dilate. A mixtured source of brain events cause vasodilation and oxygenation level increased. For both of these events, these hemodynamic parameters, nowadays we know the mechanism of the contrast agent, why there's oxygenation increase, there will be an increase of signal. It's really wonderful. We have this susceptibility signal increase when you have more oxygenated hemoglobin. This is how BOLD works. It's very potent. In our bloodstream, we have tons of the hemoglobin get oxygenized, deoxygenized dynamically. On the other side, CBV, the blood vessel volume, which is also correlated with blood flow. When the blood vessel is dilated, it reflects neuroactivities also, and even more accurate compared to the BOLD. So certain population of the researchers in the field study layer-specific fMRI using BOLD and this vascular occupancy weighted MRI methods to see the layer-specific signals in our human subject. And this is an example of a cat cortex in response to a visual stimulation. And very important message here is blood volume change and then BOLD change. They are all potent, and then they are reflecting certain brain activities. So that's solid. There are questions about in the field, right? So MRI, you're detecting noise. What's your statistics? But fundamentally, using faster acquisition of BOLD and then using the the CBV measurements, we have solid evidence that they are correlated and reflecting the neuroactivities very nicely, especially in the faster BOLD component. So they are great. The BOLD is great. It's very reliable. But the problem is if you want to see what exactly happening, the mixture source of signals, if we want to dissect it, we don't see it, right? So it's really reliable, robust readout platforms. But if we want to see Is it dopamine? Is it acetylcholine? Is it calcium? We don't. We cannot see it. So the strategy here is we use this very strong, robust readout platform, both signal, as a reporting system, and we try some strategies to link the molecular events to both signal. We call it hemogenetic fMRI. So there are several strategies, and there will be more to be developed. Depends on the target, depends on the feasibility of the methods. I'm going to introduce you three. Okay, so first of it is we utilize. So left side is about how we do it, and if you are interested in what we are detecting, the right side is what we are detecting. If we are interested in the calcium activities, one of the strategy we developed is utilize what is already there in the endogenous neurovascular coupling. There are pathways, there are calcium-sensitive enzymes already in our brain that convert the calcium activity to both signal. We use one pathway, not everyone, and then use that pathway, we overexpress it in our interest neurons to report or amplify that information. So that will become circuit specific. Second way is we generate some new links rather than use the old ones or whatever is already in the brain, because that will be challenging to differentiate. We create something, but use what the brain already have. The bioengineering philosophy is use what the biology has, we engineer it. One of this is we will generate or create pseudo neurotransmitters, which actually are responding to action potentials and can be released in the axon terminals and report through both fMRI, so that the release will be engineered to specific cell type, specific circuits. Then the readout is always bold. So the third one we are doing is totally orthogonal to generate something the brain doesn't have. The brain doesn't have is light. We don't have any light in our brain, but most of the time it's opaque, enclosed in this good quality of skull, right? So it's dark. But if we can introduce some light, luciferase have been used all the molecular assays as a reporting system. If we introduce those luciferase and link it to molecular genetic information, we can report in the both signal by manipulating our blood vessels. So one by one, I'll go through it, and some of them might not go deep enough, but you can ask questions either here or afterward. So first, how do we convert the calcium activities to bold in specific cell type and circuits? So how do we do it? So one of the target of pathways, so many pathways people now understand in neurovascular coupling mechanisms. One of them are nitric oxide synthesis. This nitric oxide synthesis is very important when people study inflammation, oxidation happening in our brain. But nitric oxide synthesis is very interesting. It's a neurotransmitter. It has this domain that binds the calcium. It's calcium sensitive. Only when calcium is binding it, it will produce nitric oxide. Nitric oxide will dilate the blood vessel potently. This is like the pathways already endogenously, and NOS is sensitive to the calcium influx. And then also, when we do MRI, we want to see, is this pathway potent enough for us to detect in both fMRI? NOS, they only occupy like 2% of cells in the rodent cortex. And when we stimulate the NOS cells, specifically using optogenetics, the level of both fMRI evoked by the NOS cells are comparable to their sensory response. When the whisker is stimulated in the sensory cortex, the black curve shows the whisker evoked level of response. NOS is comparable, just that small population of cells. Right, so this is like very good target to use to express in the other type of cells, not NOS cells, right? So what do we do? So now we generate a new pathway, a NOS-TIC pathway, just for our imaging contrast. This NOS-TIC will be calcium sensitive and can produce nitric oxide. And how does it work? We basically borrow the structure of the NOS for their calcium binding domain, the modeling binding domain. Reductase part will keep the same, so they can produce nitric oxide. The other part is a different design, catalytic part, because we want to introduce something in the brain that different from the endogenous ones. So we introduce this catalytic part from the iNOS. iNOS is inducible NOS. NOS is only detectable in peripheral system. Only when we have inflammation, the brain will start to express it, and it has calcium activities. There's no calcium sensitivity at all. It's produced nitric oxide. At the beginning, it's protective. At the later stage, it's really damaging our brain. In stroke, epilepsy, we all have this iNOS. But in healthy brain, there's no iNOS. So, so we put this part at the catalytic domain and introduce our Gnostic. So when we suppress the Gnostic, it will only suppress iNOS, but we don't have iNOS there. It will suppress itself, right? So one, calcium sensitivity, we check it in the in vitro cell culture, see if Gnostic works or not. And also check if the Gnostic inhibitor profile are differentiable from the endogenous ones. Two strategy. One is LNAB. LNAB is a suppressor for blocker for NOS. It block both NOS and Gnostic substantially. And the 1400W is iNOS blocker, developed by UT Southwestern people, actually. When I search the literatures, now I'm in Southwestern. Right, so it suppressed the Gnostic very nice, but not much for NOS. So that's a strategy already there. As long as we express it, we get the function response, we can suppress it, we can identify it. And this is effects. Oh, the resolution is low. The 1400W is iNOS blocker. It doesn't affect the normal brain function. No effects on the hemodynamics. LNAB suppress NOS. We have NOS in our brain, and it has effective suppression. So 1400W is like safe to use and can use for differentiate Gnostic and NOS. So with this, we have a platform in the lab. We always can test if this in vitro idea works in vivo or not, because we are using hemodynamic as a readout. We deliver the Gnostic expressing cells to a rat brain with 14W or not, or there's no Gnostic. In the last column is GFP. So only Gnostic cells, when they are stimulated with this calcium mycin, when there's calcium influx increased, they release nitric oxide, and we record all the way in the MRI using the T2 star both fMRI acquisition. We have observed very nice confined areas signal increase up to 15%, and they are only shown in the Gnostic cells, and then when the Gnostic is suppressed, the activity is suppressed. So it is specific to Gnostic expressing cells and detectable in MRI. To quantify it, we see the level of difference from Gnostic group is significantly different from the control ones. And then the spatial-wise, right? So nitric oxide is diffusible gas. When we check the location, actually it doesn't diffuse that much. It's pretty refined to where the cells are implanted. So when you utilize in the in vivo study, it's supposed not to diffuse that much because this is super dense cells. Okay, so then we use this probe for scientific question, neuroscience question. One of the scientific question is connectivity. We like to see which brain areas contribute to the other. Nowadays, most of the most commonly used methods in the field, resting state fMRI, correlational analysis, diffusion tensor-weighted MRI for anatomical imaging to see the tracing part. And then for some animal, we also have viral tracing, anatomical, and optogenetic ones. Cool technique, but doesn't trace monosynaptic. It basically trace the effects overall, especially when we image in the fMRI, we can see multi-steps of pathways are activated. So there's no projection, one-step projection, functional imaging. So we can use Gnostic to do it. because Gnostic is genetically encodable. We can express it in this viral transsynaptic tracing methods. And when Gnostic is expressed, when there's no functional imaging, we don't know anything. But when we do fMRI, we can check it with the blocker or without the blocker of 1400W. When we suppress the Gnostic, we get the suppressed Gnostic activities, right? So when you do the differentiation analysis, you will be able to track the drug effects, review Gnostic specific function. So that function will be circuit specific because Gnostic only express in the projection neurons. Here we are interested in stratum. My lab is only interested in stratum. That is very critical for learning and decision-making tasks. So when we do this, we acquire two images, two maps, one without 1400W, without blocker, with blocker. And then we get a differential result in the Gnostic group, in the control group. So you can see the Gnostic specific group has these clusters of signals appeared. So they are different from the normal MRI. When you have this, we have is reward stimulation. We stimulate the dopamine neurons that project from the midbrain to the stratum, stimulate the axons. You can see the widespread brain activities, but Gnostic specific signals are only located in few clusters. And then we check the ROI analysis because the level of noise is quite significant in the MRI. And the ROI analysis brings us more confidence, see the pair comparison about the blocker effects. The 4 brain areas we observed are the projecting brain areas that activated during this reward electrical stimulation. And the time course is also very interesting. Is the blocker effects you can see suppressed the overall time course, and with the 1400W, the signal the signal is much lower, but the without 1400W is also not extremely different from the endogenous ones. So the profile, the temporal behavior of this Gnostic is similar as the endogenous ones. We confirm further, like all the tools to confirm. Are they really reflecting neuroactivities? One of the ways to do is CFOS staining. CFOS is immediate early genes. When a neuron is activated, they will be expressed immediately within this half hour to 1 hour windows. And by checking the overlap of the CFOS expression and then the mCherry. mCherry is a tag we put in the Gnostic. So mCherry shows where the Gnostic is expressed. CFOS shows where are those neurons are activated. So not all the anatomically projecting neurons, like what we see in the video here. We don't see it right here. This is a cleared brain video. Let me see. Okay. Right, so this is a cleared brain images. So it shows the mCherry signals. That's anatomical tracing. Not all the anatomical tracing represent the activated brain areas. So when you see the orange color are the real activated, that's where the Gnostic probe is targeting. So we show the functional connectivity. And then the functional connectivity signals are correlated with the cell density that express the NOS. Right, so the level of expression, the cell density are linearly correlated with the signal we are reporting. Right, so again, this is the first methods that we can actually review in fMRI, a active synaptic input to our interest brain region. And then currently, our lab is using these methods to study behaving animals when they are performing the reward learning task. What is the input brain regions are actually functioning during the learning stages. So second one is a new methods we are developing is how to imaging the synaptic transmission using this new pseudo neurotransmitter. And when we do it, Actually, we utilize the neuronal secretion pathways. We have all neurotransmitters, neuropeptides secreted from vesicles. We have several different types of synaptic vesicles. One of them, large dense vesicles, are the vesicles actually enclose all the neuron peptide and then several neurotransmitters mixtures. They package it and then transport it to the axon terminal as they are more mature. And then when there's a action potentials, the terminal calcium level increase, the membrane gets infused, and the large dense vesicles release all the peptide, all the proteins it has enclosed. That's how neuropeptide is released. So we use this mechanism to have the system also release vasoactive peptide. So neuropeptide is released, and something else is also co-released as a reporter. How we do it? The reporter part. In my lab and also my previous lab, we have done a lot of this visual manipulations because hemodynamic is this really robust readout in MRI, right? So we tried two types of peptide. CGRP is a peptide that we know has this vasodilation factors, and it's also a factor in our migraine disease. The other one, PCAP. PCAP is actually is a neuropeptide also in our hypothalamus, deep brain area. It has very potent vasodilation effects. When we infuse those peptides directly to a rat brain, we can detect the both fMRI signal increased immediately. And then very important is this PCAP as a EC50, this affinity to its main receptor is 0.03 nanomolar. Like very low amount of PCAP will induce blood vessel dilation. Right, so that inspired us to use PCAP as this pseudo neurotransmitter. Right, so our design idea is we will have some construct that express in the neurons. Neuron itself has its own peptide, and we have our own ones, the one with sorting signal, synthesized sorting signal, and the visual peptide. The sorting signal sort every peptide to their targeted vesicles. Large dense vesicle is one of it. So we choose the sorting signal, lead our peptide to be loaded with other peptide into these large dense vesicles. And once it's in, the sorting signal will be clipped off. So you have this cargo shipping everything to the terminal. And when there's action potential arriving to the terminal, they will release together. So this is not a new idea. People use this idea in optical field to see how the vesicles are transported. What they tag is optical reporter, JFP. We tag our pseudo neurotransmitter. So theirs is anatomical, right? So they just tag it, say, where are they? We are tagging them, are they released? Are they released? Can we detect it? So first, can they get expressed? We have our design, we check in the mouse brain, and we check, can they get expressed? Yes, are they expressed in the right location? Use a large dense vesicle membrane protein, VAMP2, as a staining target. We have the very nice co-localization. Then we check the functionality. We want it to be expressed and also released so that it can be detected in MRI. So we build a in vitro system to mimic what is in the brain. In vivo today is very complicated, so we simplify it. We only have a sensor that actually can be expressed in the cell. When the cell is activated, the sensor can release PCAP. So we call it sensing the action potential. And we have the sensor designed and use N2A is a neuroblastoma cells, very robust pre-cells, pre-neuron type of cells, has neuron secretory properties. So we have it expressed in the N2A cells. Secondly, we have a reporter. In the MRI, we will use the blood vessel as a reporter. But in the in vitro one, we pick a cells that has the PCAP receptor and can report by the biology assays. Here is a assay. We have the PAC1 receptor expressed, and then it will active the downstream luciferase as reporting. Right? So if you see this activation, then very likely in vivo it will active a blood vessels that express PAC1. PAC1R is everywhere in our blood vessels, even parasites. Right, so very high affinity for the PCAP to PAC1R. So we have this expression of our ASAP. We call it ASAP, axon terminal specific action potential driven sensor. We have the sensor cells, we have reporter cells. We put them together in one dish. Right? So we stimulate the sensor cells. When the sensor cells is stimulated, we are expecting the reporter cells generate a signal that can be detected by this optical plate reader. So what do we have here? When we stimulated the N2A cells, the sensor cells with different concentration of calcimycin, introduce the influx of calcium to the cells. We use the luminescence assay to read the reporter cell signals. There are great gradient of responses. Higher concentration of stimulation, higher response. And then the level of release is comparable to the nanomolar level. We use the same assay, just add the PCAP to see, okay, if you release 1 nanomolar, what level of signal you will get? We get 800 level. And then what's the maximum range we get when we have a calcimycin stimulation is between 300 to 400. So it's like a maybe 0.1 to 1 nanomolar range. And then the affinity for PCAP is 0.03. It's like far beyond that range. And if we release in the in vivo, which we are currently working on, we are having this real in vivo ones, have the neurons express our ASAP in their brain, and then we use optogenetic to stimulate them. Optogenetically stimulate, calcium increase. Are they able to release PCAP, and then we pick it up in both fMRI? So that's ongoing work. We'll have those reports hopefully sometime soon. Yeah. People are still working like now across the street. Okay, so the third one is beyond neuron activities. Like, there's when the brain is a function, there are so many things, the genetic levels, metabolic levels, everything supporting system make our brain function. So all those molecules, we want to link it, use luciferase. Basically, it's also not new. Luciferase are used in all those screening processes for people study cancers, study molecule signalings. We are using it just now for our PCAP1 receptor, right? So we use it. And then now we can bring this dish to the brain. We have the luciferase expressed with a molecules we are interested and try to read it from the brain and from MRI. So for this technique, the really key part is not luciferase, but how do we read out? The readout part is we optogenetically photosensitize the blood vessels. So the blood vessels will express some small proteins. Can be proteins, can be this opsins. Those ones are light sensitive. So when there's a light, your blood vessel gonna dilate or constrict. It can be therapy topic also. But anyway, for this reporting system, once we photosensitize the blood vessels, then the leftover question is like, where's my light? Where's my target? And those are all solved in the biology field, right? So combine these two together, we are able to report by both fMRI. Both fMRI is already there also, right? So there are group people from different point of view. They are not doing imaging, but they are interested in manipulate blood vessels. So 2001, this is 2021 and 2022 papers from MRI field and then from the 2-photon field all provide or proposed ways to manipulate blood vessels. Thromboxane expressed in the blood vessels can cause constriction. BPAC, another enzyme, and active the CMP pathway will dilate blood vessels. This happens, and we can detect it in the both fMRI. When you see the blue means visual. Oh, okay. So they don't like negative bold, I guess. So when the thromboxane is activated, the negative bold fMRI was observed, and when the BPAC is activated, you see the red part are the visual activation. So in the two-photon imaging literature, they basically reviewed what is really happening in the blood vessel level. I'll play this. So when thromboxane is activated, you see the blood vessel constricted. So it's happening, and both fMRI can detect it. So that's our design. So our design is we choose the visual dilation. We don't want to constrict it for several ways, but we could also develop that strategy. Well, first step, we are using the visual dilation strategy. We express the BPAC that can dilate blood vessels and detectable in both fMRI. After we express them, we check if we can report light. Where's the light? Light is some cells that express luciferase, right? So some cells here, basic concept is we just have some cells express luciferase. There's no promoter driven, there's no events. They always have luciferase. Can we detect it? Yes. So this is a really fun experiment we did. We tried, so when you have luciferase, there's no light, you have to supply substrate. Substrate is delivered by IP or IV. When you injected the substrate, it will circulate to the brain. It passes the BBB, circulate to the brain. Only the area where they have luciferase, the substrate will emit light. So luciferase is an enzyme. The substrate is the one that emits light. It emits light, and we are able to pick it up in the MRI. So our cells here, again, we have the cells delivered to the brain. They express luciferase. We have this CTZ as a substrate circulate in the brain everywhere, but only the region where we have luciferase, they have light. The light was picked up. We did twice and 3 times in the same animal. Just waited like 10 minutes or 20 minutes interval. This circulation is happening 3 times, and then we are able to repeatedly detect it 3 times. So this is a first time, right? So a blood vessel is picking up a light that's biologically generated. And then is the light. We can control the light. We don't want the light always to be there. Luciferase is not always expressing, but expressing meaningfully. Here is neuron. We are interested in neurons. If there's this neuron express luciferase, can we see it? What we did is virally express the neurons with luciferase. In the brain areas, we like stratum. Stratum is our favorite place. And for these 3 animal examples, when we have luciferase expressed in general neuron population in the stratum, when we supply the substrate, we continuously recording the MRI signals, both signal, from beginning till like 30 minutes. And when we record, we observed there's a rising signal in the areas where we have luciferase. There's nowhere else has this rising signal. Physics increase and decrease. Time scale-wise, it costs like 5 minutes. So slow dynamics things, but report the luciferase location. And we also check the spatial-wise. So I think there's more work to be done to get the spatial localization more accurate because of the resolution. Right. So currently in the lab, we are using this technique to detect dynamic events, dynamic molecular events. One of them are circadian rhythm in our whole system. Like this is the first. This example is in 1996. People observed in the fruit flight, in the eyes, peripheral system, using luciferase, they see this oscillation of clock genes. And in our brain, actually, we have a clock gene center. Also, all over our brain, the clock. When I say clock gene oscillation, it's 24 hours, maybe day and night oscillate in our brain. We have also this oscillation. We have a clock center. We have even the amygdala neurons, the level of the clock genes, several clock genes, are oscillating. In the disorder, the brains, the oscillation are different. So currently, Ray, the students, is doing the clock gene whole brain detection to see how it oscillates, which area of the brain oscillate in which phase. So she already are able to photosensitize the whole brain. This is a mouse brain slice. She expressed the BPAC in this dense levels, right? So then her work is really get the clock gene express lucifer as well. We collaborate with Joe Takashi lab and Color. So this is will be first time we can see a clock gene oscillation in the mouse brain in vivo. Right, so wrap up. I introduce you 3 types of technologies. Hopefully there will be more in the line of hemogenetic fMRI concept. We are able to convert calcium signal to both fMRI in the awake animals, and then we apply it specifically for this stratum input system during a reward stimulation by stimulated dopamine neurons. And also introduce this ASAP concept of hemodynamic fMRI that we can target the synaptic transmission using the pseudo neurotransmitter. Haven't mentioned is now we are working on for neuropeptide specificity. Right, so we are using the large dense vesicle, which is a synaptic vesicle specific for neuropeptide. And neuropeptide like oxytocin and BDNF, those are very important ones in healthy brains and the diseased ones, but we don't have idea about when they release, where do they release. So those are the ongoing work. And also the luciferase luminescence-based MRI, we call it BLI, blush, right? So, anyway, so the luminescence-based work, we can detect neurons that express luciferase. Now we are linking it more to dynamic signals, gene expression ones, clock gene. We are also trying to target different cells like microglia when they are not active. When they are active, their signal level of luminescence, if you express luciferase, are very different. And can we catch it? And in inflammatory brains, they have this microglia aggregation. Can we report it in MRI? So this is also ongoing for the third project. So by that, I'll thank my lab. We have more lab members supporting roles, and the main 4 I listed here, and also our funding mechanisms. Also, all the work is based on my previous work in MIT Alan Jasnoff lab also. So they are also working actively. I should mention another colleague also in the academic field, Robert Ollendorf. He's also working on the hemodynamic direction. So I hope this field can actually boost, and then we can understand more fundamentally about the brain circuits, brain molecules. And as I mentioned also, when we understand more in the animal world, we also want to see how much information insights we can provide and also learn from the human imaging field. And that will be the goal. Okay, thank you.
Wow. Thank you so much, Dr. Li. That was really incredible. That was not a view from the cheap seats. You got the real deal here. This is fantastic. I want to open it up for questions from the in-person audience at first, and as time allows, we'll take some from the online audience. All right, let me bring the microphone over to you.
For that luminescence slide in the in vivo, you have the 3 expressions there. Which one's the amygdala? Is it the bottom one? Yeah, yes. You mentioned about the
amygdala is the middle ones. Right. So what I show here is the 2 temporal dynamics of mRNA and the protein levels. What we are detecting in our MRI is the protein levels. Only when it's expressed, we are reporting it.
Hi, thank you so much. That was really high level for someone like me that doesn't do imaging work, but I could really follow what you were saying, so thank you for your really clear explanation of your work. And my question is, with the 3 different methods that you're exploring, and I know it might be a little early to answer this question, but which do you think could be most translatable to human research in the future?
No, this is a good question. And also, I was asking myself also because I want to develop some tool, right? So in different stages, but if something can be used in human subject for whatever ways, so currently I still think I cannot choose, but say which one is closer. The luminescence one could be closer because we don't have light in the brain at all. And if there's light, it doesn't hurt unless you photosensitize the blood vessels. And then the photosensitization can be therapeutic goal also because the vasodilation, targeted vasodilation. We know this neurovascular coupling is a closed loop. It's not we report neuroactivity, only use the vascular, but vascular tones, if we manipulate, we can also improve or change the neuroactivities. So something can be done in the future vision-wise, but not immediately. And for the second approach, I think that could be also a potential. We're not there yet. Right? When we release something, we might release something more inert. Right, so more inert, then people are fine to have this thing. Like it's like, oh yeah, I release something that doesn't function at all. Only if I induce something in my blood. Again,
for those pseudo neurotransmitters.
Right, that's pseudo.
Method.
Right, so as long as it's inert enough, I think people are willing to having it for diagnosis and therapeutic. If you have something that can help you to see what's going on and also help you to treat, like the both, then it's, I think, more and also safe, right? So you have to be safe. That direction, we can think more about the strategies.
Thank you.
So I have a very naive question of something you were talking about in the beginning, about something you were measuring, looked at the brain's response to oxygenation. One of the treatments that people are talking about for brain injury is hyperbaric, where people's brains are given very high levels of oxygen. Do you think some of your methods could see what happens in the brain when I get a lot more oxygen or completely oxygen deprived? Do you feel like something that you're looking at could answer those questions about improving brain health?
So do you mean a way that we can measure if they do have a higher
Well, if
tissue
you
levels?
use it as a treatment, hyperbaric chamber treatment gives the brain more oxygen. They breathe in very high levels, 100% maybe, and pure oxygen, high pressure. So would your measures be able to look at what happens to neurons if you give it increased oxygen?
Yeah, I think this is a good application, right? So I heard about this therapy. Yeah, right. So if we want to examine and then see what neuroactivity is, because we don't want to contaminate with bold or other things, right? So then when we use this, we're going to be cautious about the bold this time. We might not use bold, but use CBV, right? So we use CBV to measure, and we, for example, use Gnostic, use CBV as readout and challenge with different level of oxygenation, the 100% therapy, and see the effects, the distribution of the effects across the brain.
Yeah, interesting, because the trials have been very questionable, but I wonder if maybe 20% of brains do better and 80% don't, but it'd be nice to know precision what happens. My other question is related to the clock gene. So if we're interested in healthy people's sleep, how do you think your work related to the clock gene could relate to what kind of things would we expect in people as they get better sleep in their clock gene, and could better sleep improve the epigenetics of the clock gene, maybe?
Great, this is more inspirations, right? Seriously, because right. So sleep deprivation or disruptions in circadian rhythm are gonna cause alter our brain functions. We don't know how, and then we know there are brain diseases, but also like what could be downregulated or regulated. We could use our methods to read it out. Also, in this proposal, I also proposed that we can have 2 channels, right? So if we have multi-channels to read out, then there's not just one target. We could read out multiple targets and to see what events is happening in which brain location. For yours, I think it's I really get inspired also. Thank you.
Other questions from the room here?
Do you have one?
All right, let's take a look online if there's any online questions. We have a question, if you could elaborate the basis of the bold signal, that remind people how fMRI works with that particular
Oh, great.
signal.
Yeah, so I skipped that part. I just don't want to overwhelm you with this complicated things, but let's go through it because it's such a great endogenous contrast agent. We don't need to do anything. We have it in our brain. I think this slide will be the one. So we have the bold is blood oxygenation level dependent fMRI. The blood oxygenation is all about the hemoglobin in our red blood cells. We have oxygenated hemoglobin and deoxygenated ones. And so in this slide, I have this GIF movie to show when the oxygen bind or unbind, you have these 2 states. And then in our this hemoglobin, this big protein, we have 4 heme domain. It's the heme domain that has an iron center. It bind or not bind the oxygen. When it bind the oxygen or unbind the oxygen, there is electron pairs, either paired, everything is paired, or something is not paired. If not bind, I have 1 available electrons, right? So once it's unpaired, you have 1 electron, then you have 4, then you have 4 available in the field to distort the field, right? So when you have less oxygen and distort the field, in T2 star imaging, you get a lower signal, and then it's very significant susceptibility phenomenon. So oxygenated, you have higher MR signals. Deoxygenated, you have lower. Our bloodstream, we have 1 millimolar level of hemoglobin. It's a crazy concentration we can never get for our MR contrast agent development. It's like a dream. You have such a big concentration. Every protein, you have 4 of them amplified, and then you are causing the field distortion, which is very significant, distorting the field. We can read it out robustly. So that's how both fMRI works.
So I have one last question. So optogenetics, when do you think that maybe could be applied in humans? And with the idea of more focal, because they're doing TMS and directed stimulation, what do you feel like is the future of that for human treatment of brain?
This is also a very wonderful question. So for now, all the optogenetic, a lot of manipulation stays in the basic research. When I developing all those tools, I was also thinking, how do we actually translate to human? It's a similar barrier as optogenetic, right? So we have wonderful molecular accurate tools, but for me, my vision is one of the open chance or niche that we can start is neurosurgery for people who need neurosurgery. Yes, deep brain stimulation, we could actually explore the optogenetic methods and with the invasive recordings and with this manipulation, even with more molecular accurate readout. And we can also get more fundamental readout to see what we are treating, right? So if we are deliver a TMS and then we record the electrophysiology, we only get the electrical signals. But if you deliver, it's doable. I think it's more like how we make it safe to be efficient enough, because fibers we can make as thin, even thinner than the electro arrays these days. How do you deliver it in a safe ways?
So, I'm sorry, we're at time here. We meet after the. So, lifelong brain health. This is our shared mission. Thank you, Dr. Li, for your insights and your vision, sharing your work with us. It's really wonderful.
Thank you, everyone.
Thank you all for joining us today. We hope to see you again on November 14th. Dr. Henry Banca will discuss the neural basis of cognitive enhancement from speed training.